So finally, we're going to want to do some visualizations to look at the different immune cells that we've identified. So for the first plot, we want to start off by making a vector of the order of markers. So this is going to be the order of markers that we want to see on all of our x-axes as we're going to be analyzing our data here. So generally, I put it into an order where we start with our primary markers first to look at the identification of cell types, and then can look a little bit more into secondary and tertiary markers, looking at activation level. So here you can see I started off with CD3, CD4, and CD8 to look at the different types of T cells, and then looking at some secondary markers to identify subsets of CD4 and CD8 T cells, and then some more activation markers close to the end. So the first thing that we're going to view is all of the different combinations of immune cells that have been identified within our samples. So we're using this filter for total pheno with our all FE that we have from up here. And then this is going to order our markers on our x-axis. And with this heat map all pheno, we can view each of the different cells that have been identified. So in this plot, each row represents a unique cell phenotype, where green indicates positive expression of a marker and blue indicates negative expression of a marker. So here we can see that we have a lot of cells that express CD3, but not quite as many that express CTLA4. We can also quantify the total number of populations that were identified with this in row. So this counts the number of rows for the total phenotypes. And so we can see there were 10,912 different populations that were identified in this study. Now, because that's a lot of different cells to start to look at, we're going to filter to ones of interest. So in this study, we were wanting to look at CD3 positive cells that constitute greater than 1% of the total live leukocytes in a sample. So here we can see we took our all-FE. We're filtering to those where CD3 equals 1. So those cells that are CD3 positive, and where the percentage is greater than 1 in at least one of the samples. And so now we're going to filter to our different populations, and we can see that there are a total of 14 populations identified. So this is filtered down our 10,000 populations to 14, much more manageable number to look at. Now, to calculate the number of cells in each sample with these 14 different phenotypes, we can use the function identified pers. So we're taking our sample populations here, so these are these populations, our data from our all-FE, so this is all of our data. We're using that order of markers and to get our sample populations all groups. And so here we can see for population 1 in this file, we have 2% of cells that express population 1, we have 2.7, 1.9, and 1B, 1.1, and 1C, and so on. And finally, we can visualize these flow populations. And so this heat map is going to show the CD3 positive phenotypes that constitute greater than 1% of the live leukocytes in one sample. And again, green indicates positive expression, blue indicates negative expression. And so we're taking this sample populations from before, using the function column to row names. And then we're going to take that, select all of the order of markers vector, convert our factor to numeric, and then use this P heat map. We're not going to cluster the rows or columns. And then here's the cell width and height, 15, angle column. So this is the angle down here that we're seeing. This is the colors for the green and blue. And we're not going to add a legend in this case on this plot. And so here we can see our 14 different populations and the different markers that are expressed. Now, another thing that we can do is a correlation plot. And so this compares the percentage of cells in each population to other cells in the other populations. So this can be used to explore associations between different cell populations. So we use this calc_cor function to calculate the correlations, and then superheat to create the plot here. So here yellow indicates high positive correlation and purple indicates high negative correlation. So here we can see that, for example, population 1 and 2 are fairly highly positively correlated with each other, whereas population 1 and 13 are highly negatively correlated with each other. So for example, as population 13 increases, it seems like population 1 is generally decreasing. So in this study, we had different time points. So we had day 30, 60, and 90. And in this case, we can create these time series plots to compare how each of these 14 populations that we identified have changed over time. So this is using just normal ggplot functions to create this plot here. So here I'm taking the sample populations all groups. So this is the information that contains these 14 populations and the percentage of cells in each of our samples that express each of these 14 populations. Here I'm just cleaning up the data a little bit again. So I'm separating our file name into their separate time point group and the mouse number. I'm grouping by all of these and finding the average percent for each of our groups. Cleaning up the names a little bit here so anywhere where it says group 1 is our control group, anywhere where it says group 2 is our vaccinated group, and then converting our time point to remove where it says day 30, 60, and 90, where it's just going to be the number. And then here I'm taking out just the population number, so it doesn't say pop 1, it just says 1, 2, 3, 4, 5, etc. And then here, just using a normal ggplot, so we're taking this pops for plots average, or on our x-axis, we're gonna have our time point, y-axis, we're gonna have the average percent in each of our groups. We're gonna group by our group, and the color is also gonna be our group, so we have control is red and blue is vaccinated. And we're doing geome points, so these are the points we see here, and connecting them with our geome line. And then here we're facet wrapping here by the population, so we're gonna have a different plot for each of the different populations. Scales are free so that we can have different y-axes, numbers here on the side. There are 5 columns, and we're going to label both where it says pop and the numbers here on our strip text, so we can compare the different changes in populations over time. So one of the last things that I'm going to show today is different visualizations that we can do with both our flow cytometry data and then other experimental data. So as I described in the first video, we have information in a lot of our experiments about the bacterial burden expressed as log 10 transformed colony forming units. And so this shows the number of bacteria that are found in the lung. And so similarly to the flow cytometry, we have a different measurement for each mouse. So the first thing that I'm going to do is read in our colony forming unit data. Because our flow cytometry data only looks at the lung, I'm filtering to the lung data. And our flow cytometry data also includes just our groups 1 and 2. And so I'm just going to filter to the data that matches with our flow cytometry data here. So this is just reading in our Excel file here and then filtering. And so this is a little bit of what the data looks like. We have our group, our mouse number, this is from the lung. We have our different time points, and then CFUs here. Then I'm going to be cleaning up the flow cytometry data because we're going to be joining our CFU data with our flow cytometry data. So we need to have the column names and whatever is listed in the columns be able to match up. So originally we're taking our sample populations all groups, which contains information about each of the different populations and the percentage of cells, and then similarly the same time point group and mouse number that they came from. So the first thing I'm doing is separating the file name into the separate time point group and number category, so that here we can see the time point group and number are going to match up the same with our CFU data up here. And beforehand, the time point had said day 30 or day 60, day 90, so I'm removing the D and just making it say the number. So again, that matches the time point here. And then our CFU data, the time point and the group number are numeric columns. And so I'm just making sure that they're numeric here as well by setting them as numeric. So now we can see that our group number and time point match up for the 2 different data sets. So in here I'm going to be doing an inner join. So I'm joining the CFUs, the pops for CFUs, so that's our flow cytometry data, with our CFU data. And so it's going to match up all of the data by the group, the mouse number, and the time point. And then here, the population originally says pop1. So here I'm extracting just the number. So here we have all of the numbers right here. And so here we have our CFU data and our flow cytometry data. So one of the things that we're going to do with the plots is perform linear regressions. And so we need to actually calculate these estimates and perform the linear regressions. So what I'm going to be doing is first pulling in this pop CFU, so the data that contains our flow cytometry and CFU data. I'm going to be grouping by our population here and then nesting it. So that means each of the populations is going to have its own data. And then here's where I'm performing our linear models, where we have our percentage data and CFU data. So this is going to be our model here. Doing this for each of the different populations. So each population is going to have a different linear model. Then we're going to take this model, use the map function from the per package, and tidy it up with this tidy function. So now we have our summary model for each of the populations. We can then unnest our data. So this, we're going to see all of our different populations again. We're just going to be taking our CFU term in this case, because that's going to be giving us the information about the slope of the line. And then we're going to be selecting our pop, our estimate, and our model. Then we're going to tidy the model, and we're going to use this glance function from the broom package, unnest it again, and then select just our population, adjusted R-squared value, p-value, and our estimate. And because we're doing a lot of comparisons, we're also going to apply a multiple comparisons correction, in this case, Benjamini and Hochberg, which is this BH. And so this just corrects our p-values and accounts for the fact that we're doing a lot of different comparisons here. So we have our original p-value here. We're using this p-adjust function and going to be, so this value that was 0.0000581 is going to become 0.0008131. So it's adjusting all of these p-values to account for the comparisons. One of the other things that we want to do just to make the plots look a little bit better is calculate where we're going to be placing our labels on our ggplot. So if I'm going to scroll down here to see where we're going to put these R-squared values, that's what we're going to be calculating here. We're pulling in our populations, our flow cytometry and CFU data, and our values for plot is this right here. So this has our adjusted p-values and as well as our estimate. We're going to be joining by our population, so pop column right here. Again, we're going to be grouping out by a population so that every calculation is going to be for each of the different populations. We're calculating then the average percentage. So this is the average percentage for the flow cytometry within each of the populations. And our x-axis label is going to be calculated by finding the range of the x values, so the maximum of the percentage minus the minimum of the percentage, and dividing by 2 to find the center. And then for the y-axis, we're going to place a label as the smallest y-axis value if the relationship is positive, so if the linear line is positive, and the largest y-axis value if the relationship is negative. So yeah, if the estimate is less than 0, so negative, we're going to take the max CFU value, otherwise do the minimum CFU value. And so then here we have the average percentage for flow cytometry percentage for each population, our adjusted R-square value, a p-value that's been adjusted, and then where the label is going to be placed on our x and y axis. So then when we plot our data here, we're going to pull in our flow cytometry and CFU data here. With ggplot, we're going to do a geom point where our x is the percentage, so flow cytometry, our y is the CFUs. All the dots are going to be in black. We're going to add this geom smooth. So this is that linear model here with the same percentage and CFU. And we're going to make this a blue line here. That's why we have blue here. For the geom text, this is this R-squared value. So we're going to add on our R labeling. So that's this object right here. And our x value is going to be our x-axis label position. Our y is going to be our y-axis label position. And then the label is going to be, it's always going to be R squared is equal to. So we have our R squared is equal to. And then we're going to round our adjusted R squared value to 2 decimal places. So that's why we have 2 decimal places here. Then we're facet wrapping by each of the individual populations. And then in this case, we have a free X. So the x-axis here is going to be different for each of them, but our y-axis, because the CFUs are going to be the same regardless of the population, can stay the same. And so then we have our population and CFU linear regression plots shown here. And here we can see, for example, this population 1, the R-squared value is 0.62. So it looks like as a percentage of cells is increasing, the log 10 CFUs are decreasing. So this could potentially be a protective population.